From a study tracking 500,000 people over 40 years, for over-the-counter drugs that extend lifespan have been identified. One of them is Viagra. It has been reported that drugs such as metformin, SGLT2 inhibitors, and rapamycin have the effect of extending lifespan. This time, researchers from Switzerland used data from UK Biobank, conducted a tracking survey for over 40 years on more than 500,000 patients. In this study, the correlation between the experience of taking 406 types of prescription drugs and mortality rate was investigated. As expected, the results showed that many drugs tend to lower survival rates. This can be considered as a result of drugs having a negative impact on the human body. In another view, drugs are taken to treat illnesses. It may simply appear that taking medication reduces survival rates. However, interestingly enough, results have shown that some drug users have lower mortality rates. The 4 drugs that seem to lower the mortality rate the most are atorvastatin, which is used as a high cholesterol treatment drug; sildenafil, the ingredient of Viagra, includes analgesics such as naproxen, hormone replacement therapy estroderm, and others. However, whether these drugs actually have a life-extending effect by taking them, researchers believe that more detailed analysis is needed. To give a clear example, taking Viagra does not lead to longer life. The logic that people who are prescribed Viagra are healthy and likely to live longer. By the way, this graph shows features found during the survey process related to increased mortality rates. It is observed that smokers have a higher mortality rate than those diagnosed with cancer. Being male is, being elderly and, diagnosis of diabetes and, has been analyzed to be a factor that equally increases the mortality rate. Alcohol consumption and BMI do not have much influence on mortality rates. Fasting for 24 hours twice a week activates NK cells in mice with cancer, improving therapeutic effects against cancer. In experiments where cancer cells were transplanted into mice, they were fasted for 24 hours twice a week. Fasting lowers blood sugar levels, increases lipids released from fat tissue into the bloodstream. NK cells. Fasting leads to using fat instead of sugar as an energy source. The tumor cells inside use up all the available sugar. But normal NK cells are less active in this environment. NK cells that can use lipids as an energy source through fasting can attack cancer cells more aggressively. Fasting has been found to reduce fat and enhance the effect of cancer treatment. An effect has been reported to increase metabolism, enhancing immune function. It appears to enhance the effect of cancer treatment and attack cancer cells more efficiently. By the way, a side note, despite fasting twice a week in this experiment, there was no weight loss in the mice. During fasting periods, the environment becomes less favorable for normal NK cells. This seems to be because the mice were allowed to eat freely. This is interesting from a diet perspective. Even fasting twice a week, this means that weight does not decrease when mice eat as they like outside fasting periods. Research has found that people who have been infected with COVID-19 are 50% less likely to catch the common cold. The key is cellular immunity to COVID-19 proteins NSP12 and NSP13. In addition to COVID-19, SARS-CoV-2, humans are infected with various other coronaviruses, some of which, like HCoV-OC43, are known to cause the common cold. These viruses have similar structures, but it was not well understood how effective the immunity acquired from COVID-19 infection would be against other cold viruses. In this study, researchers examined 4,900 people who visited hospitals from November 2020 to October 2021. The results showed that people who had been infected with COVID-19 were 50% less likely to be infected with the common cold, other coronaviruses. When investigating which immunity generated by infection with COVID-19 is effective, it was found that cellular immunity, CD8 plus T-cell immunity, against the conserved non-structural proteins NSP12 and NSP13 of coronaviruses affects resistance to the common cold. Infection with COVID-19 accelerates the aging of brain neurons. Senolytics, a drug that eliminates senescent cells, can mitigate this condition, at least in brain organoids. As we age, the risk of neurodegenerative diseases increases. Aging cells, recently referred to as zombie cells, are known to be harmful to their surroundings, despite not being dead. On the other hand, neurological symptoms due to COVID-19 infection have been reported. Believed to be the cause of symptoms like brain fog, a feeling of cloudy consciousness. However, the relationship between COVID-19 and the brain and accelerated aging has not been well understood until now. According to the researchers, it has been found that the brains of patients who died from severe COVID-19 infection contain more senescent cells than usual. The researchers confirmed in this study that accumulated senescent cells in human brain cell organoids can be reduced by senolytics, drugs that remove senescent cells. This reduction suppresses age-related inflammation caused by senescent cells and rejuvenates aging-related gene transcription. The senolytics used included 4 types: navitoclax, ABT737, a BCL2 inhibitor, fisetin, a component found in strawberries, and a combination of the cancer drug dasatinib and quercetin, a compound found in onions. This study suggests that senolytics show promise in reducing senescent cells and related inflammation. But their effectiveness and safety in human would require confirmation through clinical trials and further research.